Okay, so I see there are a few participants already joining. So my name is Tobias Lenz, and welcome you all to this SIP webinar. Just a few words maybe of introduction about the webinar series. This is organized by the Society for Immune Polymorphism. As I'm sure most of you know, the society that sort of tries to bring together people who are interested in immune genetics and the evolution of immune systems and immune molecules, not only in the usual lab rats and lab mice, but in broad systems and in species that nature provides. And besides the annual meeting, we also organize these webinars. This year, we plan to organize about 5 webinars that show people that have given invited talks or shorter talks in the last year's SIP meeting in Sassenheim, and essentially also maybe giving a flavor for what may be coming at the next SIP meeting in Sassenheim in November. That will also happen again this year. And today, I have the pleasure to introduce Jacek Radwan, who is a professor at Adam Mickiewicz University in Poznan in Poland. And just to give you a feeling for how he got there, he started with a PhD at the Jagiellonian University in Krakow, and then did a postdoc at the University of Sheffield, and later at the Max Planck Institute in Seewiesen. And then he became an associate professor at the Jagiellonian University again in Krakow, and then later became full professor at where he is now at the Adam Mickiewicz University. And there is also the head of the Evolutionary Biology Group, and basically researching on a number of different topics that probably cover different aspects of host pathogen coevolution, queen dynamics, as we will hear about, I'm sure, today, but also sexual selection and the interplay of those different fields, basically. And he's also very interested in evolutionary trade-offs, which I also share a lot. His work has been very influential in the work in the field of evolutionary ecology. For myself, I've been citing him a lot and reading him, and I'm sure also for many other people. And so I'm very happy that he's presenting today sort of an extended view of what he started at the SIP meeting last year. So I look very, very much forward to hear about your work on MHC genes in the Red Queen's race, Jacek.
Okay, thanks very much. Thanks, Tobias, for this very nice introduction. Thanks, Steve and Society for inviting me to give this talk. So today I'm going to tell you about something that I'm struggling with, struggling to understand. And this is about how MHC genes evolve. We know these genes for years. We know what they do, but I still think we do mostly hand-waving about how they evolve. So just to briefly introduce those of you who don't actually work on MHC. So these are genes that code for proteins that bind antigens. So this sandwich is supposed to represent MHC molecule holding antigen by means of some peptide binding residues that are responsible for specificity of binding. And then MHC molecules present these antigens to T-cell receptors, and that just thus starting adaptive immune response. So humans, we have a couple of these genes. So we have class 1 genes that are mostly involved in presenting oligopeptides that are residing in our cytoplasm, whereas class 2 genes code for proteins that present things that are outside. So what got me interested, like many other evolutionary biologists, in these genes is their extreme polymorphism. So Most populations of vertebrates would host dozens to even hundreds of variants within not only single species, but even within single populations. So this extreme polymorphism has been an immense interest to evolutionary biologists, and evolutionary biologists soon after these genes were discovered and described, started to devise various hypotheses on how this polymorphism came about. And I just mentioned the main ones. So the most influential perhaps was the were two hypotheses: frequency-dependent selection and heterozygote advantage. So I start with frequency-dependent selection. So the idea here is that because pathogens whose antigens are presented to T cells are destroyed, they are selected to somehow avoid this presentation. So there will be selection for changing, for example, sequences of peptides that give rise to this to the antigens that can be presented to T cells. And they, of course, should first of all evolve very fast to avoid presentation, but the most common MHC types. This would give advantage to rare variants. So here we have a variant that is MHC variant that's common. Parasites would evolve to change their amino acids and stop being presented, and such a parasite would spread, and this would basically give an advantage to alternative MHC allele. So that's one mechanism, and this is the cycles are kind of never-ending story. This is like a no-win game, and this kind of dynamics has been described in slightly different context earlier on by evolutionary biologist Valen, who coined processes that involve antagonistic biotic interactions like the ones that are occurring between hosts and parasites, using the metaphor or red queen from Lewis's Carroll Through the Looking Glass, the second part of the Alice in Wonderland, where the character is called Red Queen lives in a country where you have to run all the time to stay in the same place. Likewise, parasites have to evolve all the time to adapt to a host immune system, and vice versa. Once parasites are adapted, hosts need to evolve to keep up. Another hypothesis that was quite influential is heterozygote advantage, which basically tells that if you have two alleles at the MHC locus, we are able to bind wider or broader range of antigens. So, for example, we have two parasite strains that differ in the antigens they present. A1 MHC could probably be resistant to parasite 1, but variant A2 would be resistant to parasite strain 2. And if you calculate fitness over these two parasite strains, heterozygote would be fitter because it'll be able to recognize both parasites to some extent, and the multiplicative fitness would be the highest for heterozygotes. So that's one of the ways via which heterozygote advantage can arise. Similar phenomena can also occur if these different parasite strains or species occur at different spatial and temporal scale, but you still need similar kind of heterozygote advantage for multiplicative fitness for these systems to maintain polymorphism. So actually the they are very similar to heterozygote advantage. I would say that they are kind of extension of heterozygote advantage mechanism. Also, sexual selection. We know that MHC can be a subject to mate preferences. So the famous Yamazaki's mice that would prefer to mate with MHC dissimilar partners, but apparently this mechanism is not, at least not universal. So I'll focus on hosts and parasites and on these two first mechanisms in my talk today. So extreme polymorphism is just one very exciting aspect of MHC. There are two more features that are very commonly observed in MHC evolution. One is the signatures of historical positive selection, meaning that non-synonymous substitutions are occurring or spreading more often than synonymous ones, which means that new variants of MHC somehow have an advantage and are entering populations. And furthermore, this positive selection mostly occurs at sites that are already indicated on my first slide, the ones that are involved in peptide binding, so-called peptide binding residues. So that's one example from our work on spotted suslik. So that's the sequence of an exon that codes for peptide binding pocket of MHC. And if the value here is above 0, it means that we have an excess of non-synonymous substitutions. And you can see that there are some peaks. And these peaks mostly coincide with sites that we know from human model to be involved in peptide binding. And this is the picture you see in many other species as well. Another striking aspect or striking feature of MHC is long gene genealogies that we sometimes refer to as trans-species polymorphism. What we mean by that is that if you look at a gene tree of here we have another example from our own work on American and European bison. So we studied European bison and put MHC class 2 DRB exon sequences on the tree along with bisons and cattle. And what we can see is that some European bison sequences are more similar to American bison than to other European bison sequences. What this suggests is that MHC genes or polymorphism of these genes go through the speciation events such that both species inherit allelic lineages and that cause this kind of picture when you plot them on a phylogenetic tree. So that suggests that allelic lineages can be maintained for a very, very long time, or for the order of millions of years. So for example, we share some allelic lineages with chimpanzees. Right. So I already told you what hypothesis we have for MHC polymorphism maintenance. So do the same mechanism can explain these other two features of MHC? That's what I'm going to spend next few minutes to ponder about. So when you think about it, possibly both negative frequency dependent selection favoring rare MHC alleles should favor also novel alleles, because when they enter population they are rare, so parasites won't be adapted to them. And heterozygote advantage likewise, when you get a new variant in the population, it'll be rare, so it'll be mostly in heterozygotes. So heterozygote advantage should also in principle favor spread of novel MHC variants as long as they are functional. And And this actually, this has been modeled formally some time ago by in 1990 in a classic paper by Takahata and Nei. And they kind of concluded that both mechanisms can maintain genetic polymorphism at MHC, but also they can both maintain positive selection, so excess of non-synonymous over synonymous substitutions in MHC sequences. That has been discovered just a couple of years before this paper was published for humans and mice as well. So the way they modeled frequency-dependent selection, however, was a little bit artificial, if you think about it, because what they did, they kind of modeled fitness as being directly proportional to the frequency of alleles, MHC alleles in the population. So X here is the frequency as is selection coefficient. So you can see that the fitness depends almost directly on the proportion of an allele in the population. And because we know that it takes typically some time for parasites to adapt, to escape being bound by MHC, we have some time lags. So we decided we will use some framework that has been developed by a group of Rob de Beer to carry out some more realistic simulations of coevolution of MHC with multiple parasites or pathogens. So the framework is pretty simple. So we kind of imagine MHC molecule as a string of bits. So you can think about this being kind of cartoon representation of antigen binding sites, each of these bits. And these bits were matched to antigens in pathogens. Each pathogen would have 20 different antigens, and we simulated interaction of hosts with 25 species. So if there was a matching bit, it meant binding, and you needed to bind at least 7 out of 16 bits to be able to present to present antigen to T cells. So with these simple simulations, we found a few things. So one thing we found was that we confirmed results of Takahata and Nei that both mechanisms can maintain polymorphisms. Also, Rob de Beer had these similar conclusions. So here we have different mechanisms. So we have negative frequency dependent selection, which I will from now on refer to as red queen. We have heterozygote advantage. We have both mechanisms acting simultaneously, and broken line is drift. So we can see that the drift is this line here. This is the population size in which this evolution is happening. So we have these 4 mechanisms, and you can see the drift, cannot really maintain any substantial polymorphism. When populations are large, then you have some polymorphism that's transient, as you would predict from neutral theory. But both, depending on how fast parasites evolve, both heterozygote advantage and red queen can maintain polymorphism. So if when parasites evolve slowly, heterozygote advantage actually maintains higher level of polymorphism. Also, the number of alleles is pretty small. When parasites evolve pretty fast, so they're able to adapt fast to MHC, and they are able to evade MHC presentation efficiently, then red queen actually maintains much higher level of polymorphism than heterozygote advantage. So that's all fine. What we're really surprised at is that in our simulation, heterozygote advantage was no better in maintaining or in causing positive selection, so in favoring novel MHC alleles than drift, whereas red queen was very efficient and gave novel allele large advantage. So that's a recruitment probability of a novel allele, and you can see that if red queen works or if red queens works along with heterozygote advantage, most of the novel variants would spread. More so when parasites evolve fast, but still over all parameter space that we investigated, this was the case. Whereas with heterozygote advantage alone, without the help of red queen processes, just was unable to do better than drift. And so we were surprised by that, and we were thinking what could cause the difference between our simulations and those of Takahata and Nei. We are expecting that because of the time lags, maybe we can see some slightly different results for red queen scenario, but we have not expected to see heterozygote advantage to be unable to maintain positive selection on amino acid substitutions. And I think we think that the reason for that is that our simulations actually started from MHC being polymorphic. And the reason why we did that is, as I already mentioned, we know that daughter species normally would inherit MHC polymorphisms from parental species. So we assumed transspecies polymorphism, so each species would start from having several MHC alleles already there. And if this happens, then when a novel allele arises, it's not much more likely to end up in heterozygote state than the resident alleles. Because if it's enough that you have few alleles segregating, almost everybody in the population is heterozygote. So that's why we think we actually found heterozygote advantage being unable to explain this prominent feature of MHC. Right. So I think this is one reason, to ponder which of these mechanisms actually is prevailing and whether red queen processes are actually working in natural populations in the way we imagine they do. Because this is a really powerful theory that can explain at least these first two features of MHC. But there are also some other reasons that I would like to also mention in passing. And it turns out that MHC polymorphism and particularly red queen processes that are acting on it may have some consequences for evolution of life histories and basic organismal features such as time at maturity and consequently body size. So theory of life history theory tells us that if you have high extrinsic mortality, then selection should favor early maturity. So this theory was built by Jan Kozłowski. He had actually a paper in Science a couple of weeks ago where he demonstrated that this mechanism is actually shaping fish evolution on the globe. With Australian colleagues, they showed that the many fish species that we are utilizing are getting smaller and smaller, and one of the reasons is that we are kind of fishing, so we increase their mortality rate and they evolve to be smaller. So that's a very powerful theory. What MHC has to do with it? Well, the thing is that if you assume that MHC matters for your immunity, so if whether you bind an antigen or not decides whether you are able or not to rise an adaptive immune response, then if you have resistant allele, your lifetime expectation is longer, and the optimal maturation time is also different for you. You should mature later because your predicted mortality is lower. So basically, an ideal connection in the genotype is a resistant MHC allele and an allele for late maturity. And this is what we actually observed in our simulation. So again, we have scenarios with red queen and heterozygote advantage, or both mechanisms acting together. So let's just focus on these two, red queen versus heterozygote advantage. So what we can see here is that under some scenarios, when you allow life history to evolve along with host-parasite coevolution, when red queen is operating, they actually mature at double the age at which they would mature if there was no host-parasite coevolution. When heterozygote advantage works on its own without red queen, nothing happens. When both mechanisms work together, you get a little bit closer to what you see under red queen. So that's one thing. Another thing is that it really also depends on how fast pathogens evolve. So we call it formally, in our simulation, we call it mutation rate, but that's basically parasite evolution rate. So it's a summary of how fast the parasite can mutate, so how fast it can catch up with the host, but also how big its populations are. Because if you have big population, the chance that beneficial mutation will happen, it's large. So anyhow, if parasites evolve relatively fast, host parasite frequency cycles are quite shallow, and you don't have much of an effect of host parasite coevolution on life history. If you have parasites that evolve relatively slowly, so it takes some time for them to catch up with MHC, what you see is that some resistant MHC alleles go to higher frequency before they are driven down by parasites that catch up. That kind of escape their presentation via a series of mutations in their antigens. And if these cycles are long enough, it allows for the linkage between life history genes and MHC genes to build up. So you can see this effect of MHC parasite coevolution on life history basically reflects the linkage disequilibrium between MHC and life history genes. And this linkage disequilibrium requires these host parasite cycles to be long enough. So I'm showing this to tell you that it really matters how these cycles look like, whether they're long or short, whether they occur at all, because this is what happens at heterozygote advantage, right? Here, most of the heterozygotes would have similar fitness, so there'll be some randomness in how allele frequency change, but still heterozygote advantage, as I showed you, would maintain MHC polymorphism, because when some of these alleles drop out, then or get rare, then they get an advantage. But these cycles are nothing like here. They don't build linkages, this equilibrium between, sorry, this is heterozygote advantages here, right, the red. So they don't build these linkage disequilibria, and they are not selecting for longer time to maturity and for bigger body size. Right. So that's all theory of how MHC and parasites coevolve. So do we actually see this process occurring. So we have a lot of evidence for associations of MHC alleles with disease. So we know that some MHC alleles give resistance to, for example, HIV or few other pathogens. But do they cycle in the way that Red Queen predicts? Do pathogens actually really adapt to the most common MHC alleles so efficiently as the theory predicts? We don't even have much evidence that the novel MHC alleles give the advantage. So I showed you the nice theory that showed that this is what should be happening, but do we have any evidence? So these are the questions that I preoccupied myself with for the last decade or so, and I'm going to show you some results of this quest. So how should we test for association of MHC allele frequency and infection? And or in other words, how should we test for negative frequency-dependent selection acting on MHC? And what you can see sometimes in literature that people are just going to the population, checking what the frequencies of MHC alleles are, and check whether MHC allele frequency correlates with infection or with parasite load or infection probability. This is not, I mean, if you really think that the correlation looks like I just described, and this is what most people would think Red Queen dynamics looks like, so this graph modified by a review on Red Queen by Woolhouse et al. Basically depicts two alleles. I already showed you a bit of this picture beforehand. So you have MHC allele that goes up in frequency probably because it was associated with resistance. Then the pathogen that has been rare but is able to infect this now common MHC type will increase in frequency. So that's basically a mechanism that I described. The pathogen genotype that's able to adapt currently common MHC variant will spread. So when you look at this, when you take your snapshot somewhere in this area, you'll see expected relationships. So more common MHC types will be more commonly infected. But what happens next is that once this pathogen is common, it selects against this MHC variant, so its frequency goes down. And you enter this area, this green area here, where MHC variant is rare, but the pathogen that was able to infect it is still common because it takes some time for this, there is some time lag there. So here the relationship should be reversed. So I think from this consideration, the only thing that we can predict is actually that we should expect the slope between MHC allele frequency and parasite load to change with time. So this is what we were trying to test for using the long-term data set that was collected by parasitologists from Warsaw University. So there were, for years, they've been catching very common rodent in European forests, which is bank vole, Cletrodontis glareolus. And they basically dissected them and searched for parasites in their intestines and also in their blood. So our aims were just to look at the association between MHC frequency and time, and to see if at different time points they sampled these populations. So there were 3 populations in northeastern Poland, whether the slopes vary in time. Also, they were lucky to find some parasites that just enter this population. So if you have an emergent parasite, it doesn't have this history of a time lag. So we can expect that the new pathogens should adapt first to the most common MHC types. So we also, using these novel pathogens, we also try to, a novel parasite, we also try to test this. So let's look at the slope first. So there was, okay, so we type these voles for 2 class 2 MHC genes, DQ and DR. We look at their beta chain that's highly variable, particularly for DR, that forms this peptide binding pocket. So these are our 3 populations. You can see that we detected quite a few MHC variants, nearly 100. But so that's a lot of predictors to work with, but we kind of summarize their antigen binding properties based on several key parameters of these amino acids to form the clusters that we call supertypes. So that's the way field biologists try to deal with complexity. And nicely enough, we ended up with the supertypes being mostly shared in these populations, so that allowed us to do some meaningful analysis across these populations. But first analysis is actually in only one of these populations, because this is one of the most interesting patterns that we found. So that's the parasite. This is a gut nematode that occurred in only one of these 3 populations, and these populations had only 7 supertypes, so we didn't have so many points to look at the correlation, but it still was, the pattern was pretty strong. So in the first season that we looked at, the frequency in the previous season predicted the infection load in the next season. So we allowed for this time lag. We didn't look at it in the same time, but we expected these parasites are not evolving so fast. So we expected then the next time we go to the field, they should be adapted to the areas that were frequent in the previous season. And this is exactly right. So this is what you could expect. But what we've seen is actually the reverse. So that's basically, we interpret it as, we just started from the phase where parasites have adapted to variants that were common, but these variants went down, but parasites are have still not caught up. And we've seen a very significant interaction between season and the slope. So the next season, the slope got much shallower, which we interpreted as possibly parasites catching up, and then we would expect the slope to go to become positive in the following years. So I'm just about to submit grant proposal because we have another 15 years of data. So we hope we'll see this slope going up in the next years. So that's our hope. So please keep your fingers crossed for this grant application, so we can have a look at this. So that's one story. Another interesting pattern we found for this pathogen, a parasite, that was actually very rare. So at the beginning of century, there was just like a single specimen or 2 found in a couple of populations. Then it has been spreading, particularly in this population, the pink one. It has become quite common over a decade of sampling. It has been rare in other populations. It was still rare in one of them. In the other, it started to spread, but only towards the end of this sampling period. And so we expected, these novel parasites to adapt to the most common MHC types, and this is what we see. For the populations in which it has just appeared, we have not seen this pattern, but possibly this is because there was not enough time for these pathogens to adapt because they have just invaded this population. Okay, so we can tentatively tick these two things. We've seen some patterns in field in our long-term data that will be consistent with associations predicted by negative frequency dependence, so the change of slope with time and negative frequency dependence, so more common, more infected for novel pathogens. But of course, this is just our interpretation of the changes of some patterns that we see. What we are missing is parasite genotypes, right? We are assuming that parasites are adapting. What we need is to see it. So that's another part of the grant applications I'm submitting. And I think many field biologists should be doing it. What we need is long-term data. There were some long-term data published recently. There was a nice paper on meerkats and tuberculosis by Simone Sommer group, but they also missed the parasite bit. So that's the outlook for future. I'll skip these few slides because I would not like to go for too long. Now, another question that I asked earlier on. So, do we have any evidence that this kind of mechanism give an advantage to novel MHC alias? And of course, you have to be extremely lucky to see a novel MHC variant appearing and spreading. So I had another idea because we've done some work on MHC on guppies in Caribbeans, and we knew that guppies on two Caribbean islands of Tobago and Trinidad, they have kind of very different composition of MHC variant. And I had this crazy idea, why don't we take a variant from Trinidad, for example, introduce it to Tobago and see, if it will spread. So after some thinking, we decided this is not such a great idea because it would be like too severe intervention into natural system. So we decided instead to do it in semi-natural setting. So use this natural variation, but do all this, ask all these questions or answer all these questions in the lab. So we know these islands were separated for at least 10,000 of years, and when we looked at the MHC allele sharing, it's like exact sharing is occurring only for something like 1% of genes. So instead of releasing guppies from one island on the other, we collected guppies from both islands, brought them to our field station, and crossed them in aquaria. And then we wanted to look at how MHC alleles from Tobago, for example, would work if we expose these alleles to parasites from the other island. And the parasite that we worked on was Gyrodactylus turnbelli. So that's these gyrodactylids are ectoparasites of fish, but they can cause very severe infection. We know that also from some RNA-seq data that the MHC is expressed when they are infecting. There are some pathway related to MHC that are activated during the infection. So we also knew when we started this work that based on mtDNA that parasites from Tobago are different from parasites from Trinidad. So you have different lineages on mtDNA lineages on Tobago and Trinidad. We then managed to sequence their whole genomes, and you can see again some lineages that are specific to Trinidad and Tobago. When we dated this split based on mtDNA, it would be something about between 10 and 50,000 years ago. So we had actually, we could say that worms from Trinidad have not recently co-evolved with worms from Trinidad have not co-evolved with MHC alias from Tobago and the other way around. So what we did next, we crossed guppies from Trinidad and Tobago because we wanted MHC to be placed on kind of randomized genetic background. So obviously MHC from Trinidad and Tobago segregated, so we could have MHC from Trinidad on randomized genetic background or MHC from Tobago on randomized genetic background or heterozygotes for the origin of MHC genes. Of course, this could also be heterozygotes because there was polymorphism within each island as well. What we were interested in was the origin of these areas. So then the beauty of the system was also that we could control exposure to parasites. So if, for example, in our field work with voles, we could not, right? So we cannot tell if the voles that would carry heavy parasite load were infected because, okay, the ones that have not parasites, they were not infected because they were resistant or because they were just not exposed. So here we could put exactly two worms on the tail of each guppy and then trace the infection. So it was a lot of work. So that's my postdoc Carl Phillips working long hours at night, scoring the number of worms. We did it every second day. And so we had this infection trajectories, and then we kind of got the area under this curve just to take the total number of worm days that each fish was exposed to. And so what we found was that actually our prediction was borne out. The guppies carrying local MHC would have to deal with higher parasite load. Their infections were heavier compared to guppies that were carrying novel MHC types. By the way, I told you that within the same islands, of course, you have many different MHC variants. So we also checked if heterozygotes had lower load, and we have not found any effect. Right, we did some simulations as well to see if the advantage of this novel MHC alias is enough to cause positive selection. So in these simulations, we use selection coefficients calculated from the advantage in resistant, and also from some work that showed how parasite load translates into survival and reproduction. So in this simulation, we simulated a population of about 1,000 fish, which will be kind of average, maybe close to the average population size in Caribbean streams. Sometimes populations will be larger, but in such big populations, sometimes smaller, but in these populations of 1,000 99% of such populations would lose a novel alleles if only drift acted on it. But if novel allele gave an advantage that we detected either for novel alleles or for novel supertypes, the positive selection was much more effective. So I think we have actually quite nice evidence here that this positive selection novel allele advantage does work, at least in guppies. I think this evidence is actually stronger than for negative frequency dependence that I presented you for voles. So now we are left with the third features of MHC, which is long genealogies. So again, I go back to the simulations that I was showing you before. I showed you that we showed that polymorphism can be maintained under these parameter sets and under these assumptions that we've made. That positive selection, yes, but only red queen, not heterozygote advantage. How about long genealogies? Because if red queen is ticking these two boxes, heterozygote advantage is not ticking this one. How about red queen maintaining long genealogies? And apparently, what we are finding is that red queen struggles. So this is drift baseline. And you can see that basically if you assume red queen working, which is this blue symbols, actually the blue symbols, blue squares overlay with red squares, but these blue squares are just below these red dots. So basically red queen either working alone or in simultaneously with heterozygote advantage, it just does not maintain allelic lineages for a very long time. And unless parasites evolve very fast, so frequency dependence is very, very tight. But even then, red queen on its own does much worse than heterozygote advantage. So I think this graph that was actually produced by Tobias Lenz some time ago gives you the idea of what's happening under red queen. So yes, you have these negative frequency dependent cycles. So when allele becomes common, its fitness starts to decrease, which means that its frequency will go down. But then when it's rare, it regains the fitness and becomes common again. So the frequencies will be cycled. But every now and then, positive selection favors new alleles that enter this pool, which kind of push the old alleles away. And this is why you see red queen performing so poorly in maintaining transspecies polymorphism. So what can favor? Well, I mean, how are we going to reconcile this? So maybe one explanation, of course, is that red queen actually works with heterozygote advantage, and maybe under some parameter space, it can actually help to maintain this TSP. But as long as red queen is working, under most scenarios, red queen will not allow heterozygote advantage to maintain these allelic lineages for long. It will drag the coalescence times down to the level of a drift. So this is simulations. Of course, this is not, I'm not claiming this is reflecting anything like reality. This just is a way of thinking about, a little bit more precisely than just being verbal models. What we need to have a look at is what's happening in nature. And I think some of the data we collected on guppies give us some hints on how dynamic this system is. So I told you that Trinidad and Tobago were separated for quite a long time, so there's not much in terms of a gene flow between these islands. So we looked at the MHC allele genealogies on both islands, and the striking thing that we found was that actually, if you look at the clades that were supported, most of them contained lineages that were represented by alleles found only on one allele. So the orange are Trinidadian lineages, blue ones are Tobagonian lineages, the magenta ones are shared ones. I told you the shared ones are very, very, very few. So you can see that some lineages must have been evolving solely on Trinidad or solely on Tobago. Some allelic lineages actually were able to spread. So that's an area that produced a new supertype that was not present on Tobago, and it actually was quite successful. Sorry, not present on Trinidad. It was quite successful in spreading on Tobago. So you have this whole new allelic lineages spreading since probably after these lineages have been split. So this does not look like a scenario that would lead to the long-term maintenance of allelic lineages. So I think it kind of supports that at least in some systems, the system is as dynamic as our simulation suggests. So what can cause this transspecies polymorphism? We think that perhaps, at least to some extent, it can be caused by novel allele advantage actually favoring introgression of MHC alleles from different species. So you probably have heard that we imported some MHC alleles from Neanderthals. We get something like between 3 and 5% of genomes, and many of these genes are immunity genes. And most notably, we have some MHC alleles from Neanderthals. We also discovered similar phenomenon in newts. So these are two species of newts that diverged 4 million years ago. Yet they apparently in the areas where they form so-called hybrid zones. So these are good species, but sometimes they are able to form hybrids that have low fitness, but sometimes they can produce progeny and apparently close to the hybrid zone, the sharing of MHC alleles between these two species is high. So apparently there is some MHC introgression going on. So we were wondering how widespread these patterns are. So this is the final story I'm going to tell you about. So that's work led by my long time collaborator Jacek Radwan, who compiled a number of samples and data from various hybrid zones of different vertebrate species. And what we looked at was whether MHC flows through these hybrid zones more easily than neutral variants. So we basically tested two predictions. So normally you have this zone when two species come into contact, they may form these hybrids, and then because selection would act against these hybrids. The farther you go from the zone, the more rare the variants of the other species are becoming. So this would be a typical steep hybrid zone, and we predicted that MHC would be able to flow through this zone, making these clines shallower. Also, we predicted that if you look at MHC ancestry versus genome-wide ancestry, again, if you go away from hybrid zone, MHC should show a shallower slope compared to genome-wide average. And this is basically what we've seen. So this is the first prediction. MHC clines tended to be wider in most of the hybrid zones that we looked at. The average across species using phylogeny into account was significant, and the same for the second prediction. So perhaps transspecies polymorphism gets some help from this introgression, which we think may be actually a result of novel allele advantage that favors MHC alleles from different species in a similar way as it favored MHC alleles from different island in guppies. So that's a summary overview of my talk. So we showed that perhaps emergent pathogens adapt to the most common MHC alleles. I told you that we missed the parasite story, but at least temporarily we can treat it as a support for red queen dynamics working on MHC alleles. We also showed that adaptation of pathogens to local MHC alleles gives novel alleles as a selective advantage, at least in our guppy system. We need probably more evidence from other species. From our theoretical work, we show that red queen, but not heterozygous advantage, can drive positive selection if we assume that transspecies polymorphism is a rule, and you normally start with MHC polymorphism and not from MHC monomorphism. But we found that this red queen struggles to maintain TSP. Perhaps a solution to this paradox is that MHC introgression is actually favored by red queen dynamics. But we definitely need more data on MHC long-term interactions with parasite infectivity genes. So I'd like to stop with acknowledging my collaborators. So there were more in the work I described. So I just listed the most, the ones that contributed the most to the work that I just described. Jacek Radwan, first of all, Maciek did all the simulations, or coded all the simulations that we designed mostly with Jacek as well, and National Science Center funded most of this work. So I'm going to stop here and thank you for your attention, for listening to me, and I'm happy to take any questions.
Thank you very much, Jacek. Yeah, I don't know how you can applaud here, but consider me applauding. Very nice work. I guess I forgot to mention at the beginning that people can, of course, put questions into the chat or into the question answer section. Either way is fine, and we'll just read them if they come in. That's clearly a great overview of many years of work, and it's nice to see how this fits together. And I think some of the questions you've been pondering about are also very much at the heart of many of us, of our research. Maybe I can start with the question that one would be a more concrete, direct question to the work on the vole, the long-term timeline of voles and parasites, where you show this change in decline between the parasite and supertype frequencies. What you did not talk about is whether the supertype frequencies also changed over the years, or whether they made somewhat stable. Can you elaborate on that?
So what we have seen, so I haven't showed these graphs, but what you can see are like patterns, going up and down. But there are mostly, most of the supertypes were maintained, at least from this, dozens of years when we are able to trace them. I hope when we get another 15 years of data, maybe we'll see some more obvious trends. Just on 4 time points, there was not much in terms we could do in terms of looking at how these frequencies changed. The other consideration is that we work with a system which is polygenic, so it's not a single locus. So I have not mentioned that, but these loci are duplicated and diverged, and we cannot actually assign alleles to loci. Okay. So that makes changes the tracing the changes in frequency a little harder. So what we are trying to do now is just to characterize haplotypes, because then it'll be easier to see perhaps some haplotypes going up or down in time compared to when we just look at supertypes or MHC alleles. Yeah. But yes, that's something we are going to look at again more closely. We have not seen any strong patterns of some variants going up and down, but because we see something like 20 supertypes and maybe 80 alleles segregating in this system. It's not probably not so easy. I mean, probably parasites would have to adapt to several types simultaneously. So it may not be just an increase in the single supertype or single allele. Also, in Simon and Sommer group, they actually observed one variant to increase consistently over 30 years. Maybe we just haven't looked for long enough time.
I was in connection or in association with the tuberculosis, right? This Mercat story.
Say again.
I was in coevolution with tuberculosis.
Yeah, right? It was. Yeah, the variant was actually also associated with increased resistance to TB.
Yeah, where TB, I think from humans is known to be very slowly evolving, so that might fit to this basically long-term trajectory. Right. I guess my next question would be sort of in this direction that when we think about red queen dynamics, it's easiest to picture the simple case of one pathogen and then one host species, and then they co-evolve, right? But then in reality, as you say, it's more complex, right? And most host species are infected by multiple parasites that they have to deal with. Then the parasites often are not specialists. They infect multiple host species. What are your thoughts on this? How this will change the dynamics you've been sort of exploring?
Yeah, so maybe I should go back to my presentation again to show you one slide that kind of let me just go back to the slide again. Okay, so this one, for example. So can you see it now?
Yes.
Right. So that's actually a simulation of a complex system with a number of parasite species, and each species may have like different selection coefficient on the host, because that will be also depending on a complex dynamics on where species of hosts and parasites sit in kind of epitope space. And what you can see here is that actually when you look at this, particularly when parasites evolve very fast and you have many of them, you don't see really strong there are some cycles, but they're very shallow. And many MHC alleles would be maintained in such a system, like maybe 80 alleles. But in terms of testing these dynamics, it's a nightmare, right? So when you go from simple prediction of a single parasite, a single or like single parasite and a couple of MHC alleles, you think about these frequency-dependent cycles. This is what we typically think about, right? In complex system, it may be shallower, it may be a little bit more chaotic, but you still see these cycles where when the variants become rare, they are rescued from being lost from population. So these red queen processes are still very effective in maintaining MHC polymorphism.
Okay.
It's just that I can't imagine how exactly to test for this cycling because they are so shallow. So one way of testing it would be just to do some intervention, for example, make some areas more common or probably more common, right? So you expect parasites to adapt to them and for them to go down. Another intervention would be just to introduce a novel parasite and see how it behaves. But actually, in a system like that, most MHC alleles would segregate and at rather similar frequencies. So I think driving some of these alleles up would be a nice experimental intervention to see, these dynamics at work.
Yeah, that makes sense. Thank you. There's a question in the question and answer section by Curtis McMurtry. I think I have to read it loud. You have modeled how peptide binding influences red queen heterozygote advantage dynamics. How do you model or think about fixed sequences like HLA-C1, C2 to cure interactions on the MHC molecule? So when sort of the cure also being host component, not co-evolving, I guess, how those interactions would affect MHC evolution.
So you mean, for example, the MHC molecules that are responsible for, missing identity signals that would not be that polymorphic, right? So we have not modeled it, but I think it was actually because they interact with keys, right? This is the ones that we talk about. And I think, there were models by other groups that looked at that. And they found, because it's kind of intriguing because these are kind of self-identity signals, so that are recognized by tiers. So they should be conserved, but they are not. And the reason why they are not is that probably because the parasites evolved these decoy molecules that pretend MHC, so they are not destroyed. So the keys, basically inhibiting keys would recognize them, and that kind of selects for these conserved MHCs to change a little bit, so to move away from these decoy proteins. So it has been modeled, and it can, these systems can maintain a degree of polymorphism. So I think, there are models of these systems by other groups.
Okay.
I won't be able to tell you the name off the top of my head. I'm really bad with names, but if you email me, I'll send you a paper straight away if you're interested.
Good. Thank you. I don't see any further questions. So I'm sure that Jacek would be happy to receive questions by email too, if you are still thinking about them. And otherwise, then I think we can thank Jacek again for this excellent presentation. And